हाँ ये आप रिकॉर्डिंग
अच्छा okay. So you can all right. अभी शुरू किया क्या उसने? So out of the many accessions which we have brought from different parts of the country, each accession has been grown into 3 rows, and out of the 3 rows, one row was retained and pruned, and remaining 2 rows which you can see in front are the pruned ones. It was pruned in November 2006. The unpruned ones have come to fruiting, but the pruned ones have not come to flowering. We are expecting flowering within 15 to 20 days, because it seems that it has reached to the stage. The indications on the top of the meristem gives a signal that the onset of flowering. The most interesting thing what we have observed is the whole Jatropha carcus, which is monoecious in nature, is trimonoecious precisely, is evidence of very much of variability. We could identify only 16 different morphological types. It's a gap between these two, and if you see it from top, some of the plants look more green, and majority of the plants are yellow in color. It is because of hypoxia, more of moisture, that's why less of greenness. It's all yellow in color. We move forward, the same material with raised and sunken bed, you can very easily see the effect of high moisture on the plants. But the line which is extreme with and full of moisture, few rhizospheric variation, this particular patch is having literally less of moisture, that's why it's green in color. And the extreme most on the divided field, which is having less of moisture, looks lush green. The plants which are looking yellow, they either have not come to flowering. This all occur because of very high moisture in it. So this very clearly proves that the cultivable land with high rainfall areas should not be used for planting purposes, otherwise it won't give you the expected results. Same is the case here in this particular situation also, where the material has been treated with gamma rays, and we have found many morphological variations in this particular population. I'll show you some plants which show taxonomic variations within this treated population. The material was treated with gamma rays 60, 120, 180 and 240 GY, and it has given a typical variation, and that typical variation can be observed by having variation for its leafiness. The tip, you can see it is more pointed, otherwise you see obtuse. There are more number of 5 to 7 lobes in it. The other plant what I am going to show you is a sectorial chimera, and it is showing its higher intensity in anti-clockwise direction. This is the control branch. You can say it's a non-mutant, and this is one with some degree. The other one, the next one on anti-clock is the one which is having higher intensity of color variation, whereas again on the extreme, that is about 10 o'clock, you don't see any variation within it. The other plant which I am going to show you is very clearly the taxonomic variant, which is having its leaf, the leaf variegation you can observe. It's a leaf variegation. it's a mutant. The other branches are not. It's a sectorial chimera which we have identified in this particular plant. This plant, what you're seeing is another taxonomic mutant in which the leaf margins are wavy. This particular character is not observed in all 542 collections with 20 plants, minimum 15 to 18 plants in the population. So it's a very big number, out of which the induced mutations have given some of the types which are not observed in the normal population. The other plant which I am going to show you is having, it's all petiole. This is normal, and the petiole color is light. You can see tinge on the petiole connection to stem, whereas here you can see the whole petiole is reddish in color. So this particular plant is having both the branches. It's having non-mutant and mutant sectors. Similarly, you see some plants in which you see the whole plant is having reddish petiole color. This is another plant of interspecific cross with purple flower, more of male seen now, but few female flowers which have been tagged. This is another interesting thing. It's an enemy. You can see some of the plants which are affected by the Weber, and insect enters through the meristem tip. It's called top borer, and it damages the stem. The other plant which I'm going to show you is having it's a synchrony. It's not at flowering at present, but this had borne 2,262 male flowers, but unacceptable female flower number, that is only 33 out of 2,262. So this change in the proportion of male versus female is actually responsible for yielding ability in this particular system. And this particular character is controlled by temperature variation. If the hot humid conditions prevail, you get a better proportion. Otherwise you get variation in it, and with the switching of high temperature, the sex switching occurs. This system is normally protandrous in nature, but with the rise in temperature it turns to protogyny.